ต่อไปก็จะเป็นเซกชันที่2นะคะก็ปาล์มจะขออนุญาตรันเป็นภาษาอังกฤษนะคะ Next session we would like to invite to talk about a novel and reliable sensitivity testing moving from biological plausibility to clinical efficacy by our guest Mr. James White is the CEO of KBMO Diagnostics in Massachusetts, the company that distributes the food sensitivity test. While have almost 25-year experience in the diagnostic and pharmaceutical industry, working global to launch a range of innovative products. He holds a bachelor degree in economics from University of the West of England, Bristol. Please give a big applause to Mr. James White.
Thank you very much.
Okay, okay, Grace. Let's start by telling some a bit about your test fits and updates researches.
Thank you. So what I thought I'd do is just give you an overview on food sensitivities and then talk a little bit about how our test works and helps in that whole area. And as the title suggests, move from biological plausibility to clinical efficacy, which is a bit of a mouthful. But really what it means is we're going to show you some evidence on why the test works well versus some of the others that were highlighted on the previous talk. So again, what we're trying to move as an organization is away from having the odd example of how the test works to more clinical outcomes. And so we'll talk about those as we go through. So again, in terms of when we talk about food sensitivities, it's a large area. It's growing, and there's a number of drivers to that. Firstly, the quality of food that everyone is eating these days, and the quantity of food. So again, if you think about the amount of processed food that is being consumed all around the world, and that's having an impact. And as we saw in the previous speaker, the impact that colorings and additives are having. And so again, it's important when you run the test to make sure you're looking at colorings and additives, because unfortunately that's probably what most of the patients that you're seeing are eating, so it's important that you test for that. The other reason it's very difficult to diagnose is because the types of symptoms are many and varied. And then the other concern there is that not only are they varied, but the time in terms of from when you ingest the food to when it can have an impact is up to 72 hours. So if you think about that, for most of your patients, it may be a struggle to tell you where they were 72 hours ago, let alone necessarily what they ate. So it's a very important, really, in terms of why this is such a good area to test for, is because of that 72-hour time delay. And we'll talk a little bit about our multiple pathway approach and how that really helps in terms of zeroing in on the key foods of interest and eliminating some of the false positives that have been associated with certainly IgG-only testing and testing like the LCAT. So again, we've gone through some of the definitions, so I'll keep this brief. But we're really focusing on the middle one here, which is food sensitivity. So food allergy is IgE. It's an immunological response, but it's typically, the symptoms are much more severe and typically very straightforward to understand which food it was because it happens instantly. With what we're looking at, as I mentioned, it's that time delay. It's still immunological, but it's that time delay that sets it apart. And equally, the symptoms aren't as severe, but clearly they're pretty debilitating if you think of something like IBS in terms of the impact that that has on people's quality of life. So again, this slide just helps talk about when providers should use the test. That's always a good question in terms of when should I use the food sensitivity test. The medical director that I have as part of the company is a gentleman called Dr. Joel Evans. Joel is on the senior faculty of the Institute of Functional Medicine, and so some of you might have heard him talk about the hormones, and he leads that part for the Institute of Functional Medicine. He also runs a OBGYN clinic in Stamford, Connecticut, which is just outside New York, And so this is what he sees when he uses the test in his clinical practice. So patients who aren't feeling well, autoimmune symptoms, brain fog, fatigue, clearly digestive gut issues. And as I say, he sees breast cancer a lot and also other cancers. So as you can see, a number of varied reasons why he uses the test. Why he uses it, and again, why I think is another important element for all of you out there, is the immune complex issues and inflammation that can be associated with foods are an underlying problem for all of the conditions he sees on a daily basis. So again, I think inflammation is a well-known issue, and I think most of the patients that you see will correlate with the fact that inflammation is a negative thing. And so that's really what he's coming out with in terms of the statement he's made. So again, when you look at this chart here, what we're trying to highlight really is how the test is different. So the food passes through the tight junctions here, and typically if you look at the kind of what I would describe the upstream area, you're looking at exposure. So you're looking at, were you exposed to that food? And that's where IgG 1 through 4 typically sits in terms of the whole process. It's an important part of the process, but as you can see, it's only a partial part of the process. What we're also looking at is this downstream area here, which is the measuring of the immune complexes. Now, the reason that's important is because that's where you're triggering whether there's inflammation or not. So when you look at the chart here, we've tried to simplify here, what we're measuring is that complement activation via the marker C3D. And that's why we're very different from just a standard IgG test, is because we're not just measuring exposure, we're measuring which foods cause your patients an inflammatory response. And so that's the big difference really when you compare it to IgG on its own and IgG plus complement. It's you're looking at two different elements within the immune system. The other point just to mention here, when we talk about tight junctions, and we'll go back to it, I know Dr. Art mentioned it earlier with zonulin, but we can talk a little bit about our zonulin test and how we've partnered with Dr. Alessio Fasano, who was the gentleman who really discovered zonulin and its impact on health. So again, just to show that this has been around for quite some time, this paper was done in December 2006, and as you can see, what it's really highlighting there was an intact intestinal permeability barrier, is therefore critical to normal physiological function and prevention. And I think in the last 10 years, there's been an explosion of papers linking healthy gut to healthy mind and basically healthy overall health. So again, when you talk about intestinal permeability and its importance, clearly food allergies and food sensitivities are an important element, but as was highlighted in the previous talk, there's a number of other systemic issues going on as well. But certainly if you consider it for your patients, eating is something they do at least 3 times a day, so it's clearly the important element of what could be causing that permeability issue is what they're eating and the impact that can have. But as I said, it's been highlighted in the previous talk, stress and some of the other elements that are also playing a factor in that. So again, what's wrong with having increased intestinal permeability? It leads to disease. And so again, as I mentioned earlier, that's one of the things that this has been highlighted in the literature. We pulled out some of the diseases that are being associated with that. And as you can see, there's an underlying element of inflammation caused by all of these. So again, further evidence based of what we're seeing and why it's so important to measure inflammation versus just the exposure. Here's some of the papers as well that it's been linked to as well. So again, to understanding and testing for intestinal permeability and food-induced inflammation is critical for your practice. So again, back to linking overall health with measuring inflammation and linking it back to your food. So again, what I thought we'd do is just look at a couple of the papers that have been in the literature in a little bit more detail so you can see how that's working. So here's a paper on IBS, and this was, again, just looking at IgG. And I think what you'll find interesting here is clearly there was an improvement, which is always good. It was a 10% improvement 26% if they'd been fully compliant. So think on about that when you come back to what we're doing with the food inflammation test, in terms of how important it is to come up with the appropriate tools to make sure that a patient can be compliant, because that's clearly a huge gap in terms of 10 to 26%. So we'll come back to some of the things we've worked on to make sure we can improve that compliance for patients. This paper just talks about IgG4 as a sub-element of IgG in terms of how that had a beneficial impact as well. This is another paper which talks about obesity and the tight link there with IgG and CRP. So again, as you'd imagine, what we're seeing is when you've got elevated CRP, which is obviously a general inflammatory marker, how does that then impact with IgG as well? And I guess no surprise there that there's a tight link between inflammation and elevated IgG. This paper talks about migraines. And again, we've got a number of examples of providers that I work with who've given me some amazing stories where patients have had migraines for many years, and that's improved when they've eliminated the foods that were causing them an underlying inflammation. And so, again, it's a very debilitating thing if you end up with severe migraines on a regular basis. And so, again, encourage anyone who's seeing patients who are having migraines to make sure they run the test, given the very impressive outcomes, not only in this paper, but in terms of what we've seen in clinical practice as well. So again, here's another one just talking about Crohn's. So again, underlying inflammation or overlying inflammation in this case, and it just shows how the significant reduction in the symptomology here as well. So again, in summary, IgG testing, again, it's demonstrated the clinical utility, as you can see from all the papers I've shown you. Testing obviously improves with adherence. So again, if you can get that compliance right, then you'll get a better clinical outcome. And again, there's been good links in terms of the elevation of IgG with underlying inflammation as well. So let me go on and talk a little bit about how we're different and why we see what we're doing as an important element for you guys to think about as you look at your practice on a day-to-day basis. So what we're really trying to demonstrate here is our multiple pathway approach. So what we're looking at here is not only just measuring the IgG 1 through 4, but also the complement activation via the marker C3d. And so, as I say, it's important because it's helping us zero in on the key foods of interest. So typically what we'll see is 5 to 15 foods per patient. And so from a compliance standpoint, it's an important word that I'll keep coming back to. It helps the patient be compliant if you're not overwhelming them with too many false positives, which is typically what you see with an IgG test. So when you're giving them a number of foods that we know are actually positive, then that's been a very helpful thing. So a little bit about the company. It was founded by a gentleman called Dr. Brent Dorval, who is my technical founder and partner today. Brent's claim to fame, which is pretty significant, he invented the first rapid HIV diagnostic. So again, the reason I mention that is because underlying the company in terms of our company DNA, as it were, is an infectious disease background. So in terms of how that then transfers through to what we do on a day-to-day basis, for example, we run 15 standards and controls per patient, and we run the test on 3 ELISA plates to make sure it's highly accurate. So again, for us, it's very important that we're giving you a very reliable and reproducible and accurate and sensitive test. And that's based on, again, the infectious disease background of Dr. Dorval in terms of how we set the company up. It's a patented test. So again, it's unique to us in terms of our ability to measure the IgG and complement at the same time. And as I mentioned, it's manufactured under ISO and FDA guidelines, and then it's run in our high complexity lab in Boston, Massachusetts. So the great news is for anyone who's done the test, we thank you greatly for running the test so far, and hopefully there'll be others joining us who'll be running more tests. They're sent to Boston, and Art can talk to you a little bit more about the turnaround time and how that's worked so well. We also own the C3d cell line, and we've developed lyophilization techniques to develop individual foods. So again, the reason I tell you that is because that gives us complete control from the manufacture of the plates to the running of the test. So we own all of that all the way through. So all the way through the value chain, we can have exquisite control of how we run the test and improve on its accuracy. Copper Priest and Amvisa are a couple of examples of where we're working internationally. So we have got a clearance to work in Mexico as well as Brazil, and probably another 15 or 20 countries around the world now are adapting this testing. A little sideline: we just recently launched in Germany, and the feedback from the German market was we're excited to find that there's a company out there that's managed to eliminate the false positives that we were seeing with our IgG-only testing. So again, we'll go through this overview in terms of the next few slides. So this is, again, we won't test you because it's not an eye test, but there's a lot of foods up there. It's 132 foods, colorings, and additives. And as I mentioned before, it's very important that we're looking at those colorings and additives, and we'll show you why. The test, as you will be aware of, is a finger stick. So again, that makes it extremely convenient in terms of for us shipping between Thailand and the US. You don't have the limitations of a whole blood sample. So again, when that card is dry, then that gives us, again, an ability to ship that over a month. Obviously, you'll know that we're getting your results much quicker than that, but that just tells you how stable the sample is in terms of what we're running. Also, on we can drop ship to patients, and we have a video which shows them how to do that finger stick. I think here in the room today, you're fortunate there's an extremely talented nurse who's going to do that for you if you take the option of running a finger stick. But yeah, that's just an example of how that works. So here's obviously a top is a good one. Below is not what we're looking for. It's a very straightforward patient report. Clearly, as you can see, red is bad, green is good. So I think that's a pretty much universal metric there. As I mentioned before, we're measuring colorings and additives. The reason why is because, as again was highlighted in the previous talk, it causes a number of issues in terms of not only from your microbiome, but just, again, that gut permeability. And aside there, the red dye, for example, that you see in a lot of foods, the processed foods, was invented originally for coloring plastic after World War II. So again, there was no concept that would find its way into the food chain, but here we are today with it in the food chain. So again, no real mystery why that's having the negative impact that was highlighted before. So again, the patient report also measures, if you can see here, Candida. So again, the great thing about this test, it enables you to look at leaky gut as well as food sensitivity. So in our mind, if you have more than 10 plus foods that are elevated, that's probably indicative of leaky gut. If you've got Candida, as we've seen that we measure, if that's elevated, that could be as well indicative of leaky gut. And then we've also added a zonulin assay, which again, back to the discussions earlier, is another indicator of leaky gut. We've also, back to the compliance point that we mentioned earlier, we've added a couple of key compliance tools. One of those is we have a dedicated personalized meal plan for each report that we hand out. So that way for the patient, rather than saying the bad news is you can't eat these foods, what we give them is some hope by saying the good news is here's some ideas of what you might want to eat as well. On top of that, what we've also done is the only thing I would say that all the patients around the world have in common is that they all have a smartphone. So what we've done is we've put their results on their phones. So again, it gives them an ability to, if they want to be compliant, to have their results on their phone and their meal plan on their phone as well. So typically we delay that by 14 days between the provider getting the results, and then there's a 14-day delay before the patient gets their results on their phone. So all we need from the patient is their email, and then, as I say, there's a delay, so you can make sure you've reviewed the results with them, but we then send them that. But as you can imagine, if they've got their results on their phone, there's a much higher likelihood that they'll be compliant. The other good news is with their meal plan there, if they're grocery shopping, they can then look at that list of foods. And the third, most important for everyone in the room today, is that it's an unbelievable referral tool, because as you know, when you go out for dinner with your friends, everyone brings out their smartphones, and they'll be saying, well, why are you not eating this certain food? And they'll show the report and say, oh, I saw Dr. X or Y, and that's the person who did this report. And again, it gives you an ability to kind of test and hear from other friends and family about the test and its potential. So again, we'll quickly go through the zonulin test in terms of how that works. Again, given the leaky gut element, you can see there's a number of disease states that it's linked to. The neat thing about it, it's a reversible protein. So if you test the patient up front, down the line, when you want to retest them, then again, because it's reversible, if they've been compliant, then you'll see that there'll been hopefully a return to the normal range of zonulin from maybe an elevated level when you first tested them. In terms of, again, some of the papers showing the benefits of it or where it's been linked to, again, and some of the disease states that have been linked with zonulin as well. This one we just highlight because I know you can get the test run in stool or serum. Just to mention that all the initial seminal work that was done by Dr. Alessio Fasano was done in serum, and that's why we've chosen to run our test in serum as this data suggests. So again, just another paper here on zonulin which talks about some of the benefits of it. This was from 2017. So again, there's new data the whole time coming through the negative impact of having elevated zonulin. So again, it's an important one that you may want to test. Again, one of the things that we're very excited about is we're launching later on this year is a next generation zonulin assay. We're working with Dr. Alessio Fasano on that, and so again, by hopefully the autumn, we'll be able to kind of share that with you in terms of the improvements. So as I mentioned, the clinical studies, we wanted to kind of give you a quick overview of what we've done. This study we ran, which was a kind of a prospective one, just looking at 30 patients, 11 months apart, so they'd run 2 samples. On the left, you can see high levels of inflammation in terms of when they initially got tested. On the right-hand side, you can see a dramatic reduction in that level of inflammation. So the providers were very excited about that. But way more importantly, on this slide, what you can see are the symptoms. So we saw a dramatic reduction in the symptoms as well. So again, we're excited to say not only does the inflammation come down, but then the corollary of that is that you also see a dramatic reduction as well in terms of the symptoms that were presented. And this list speaks to what we were saying earlier about there are many and varied symptoms that are associated with food sensitivity. So we looked at, that was a kind of look-back study. So we've just completed a prospective study looking at IBS. And as Art said, it's a big area, it's a big issue. And what I highlighted on the bottom here is it's big in the US, it's big here in Thailand and Asia. Interestingly, in the late 80s in Thailand, they did a study which showed 4%. When you look at Malaysia and other countries in Southeast Asia, what you see, the studies that were done in the 2000s and the kind of mid-2000s, that has jumped to 8 to 15%, and higher is up to 30%. So again, it's a big and growing problem, linked, no doubt, to the amount of processed food that patients are now consuming, and that I'm sure has dramatically increased, unfortunately, since the early 80s or late 80s. So again, the study that we ran was with a Dr. Esselfy in Southern California, looking at what are the benefits of the FIT test for patients with IBS. There was 100 patients, and again, as you can see from the third point here, we wanted to make sure they were definitely serious IBS patients. So again, we were very excited to see the results when they came back. We saw a 126-point reduction in their IBS severity score. We saw a reduction in their homocysteine, which again is a general cardiovascular marker, but more importantly, a really dramatic reduction in their overall high-sensitive CRP. As you know, that's measured between 0 and 3. So when you see a 0.5%, a 0.5 reduction in that, that's a dramatic improvement. And again, as we keep coming back to that multiple pathway approach, that's what you would anticipate, is that we've seen a reduction in their inflammation by eliminating those foods from the patient's diet. Another one here, which is, again, a nice side effect or benefit, is a 50% reduction in primary care visits for the intervention group as well. And so from a quality of life standpoint for patients with IBS, that's a dramatic improvement. Whether there's an economic benefit, I guess we're less worried about that. But from a quality of life, that's a great benefit for those patients. And again, I think people often talk about evidence and then how do you compare with some of the other tests. So when you look at the comparison that we've been able to do in terms of when the ALCAT test ran an IBS study, we saw twice the IBS severity reduction than ALCAT. So that's twice the IBS severity reduction that you saw with KBMO versus ALCAT. And then when you look at the IgG, it was a 50% improvement that we saw over what they saw in terms of the earlier study that we showed. So again, I think we're very happy to be able to show this, and I'll dwell a little bit on it because it's very difficult normally to compare those different tests from your position as a clinician, and it's always tough for us to kind of highlight it, but this is a really good way of doing it. So 3 published studies and dramatic improvement in terms of when you look at the testing results from KBMO versus these other 2 competitors. So again, we're looking at the return on investment as well. So that's data that we're collecting over the next 12 months. So again, in terms of conclusion, again, it's manufactured under the auspices of Dr. Brent Dorval with his HIV background. So again, making sure that we're very much focused on quality, reliability, and reproducibility. It's patented, and as I kind of keep coming back to, that multiple pathway detection technology has really helped us in terms of to ensure good outcomes. So we've certainly helped that when you see the compliance tools that we have, and those are being shared now all around the world in terms of the meal plans and also the patient app. And then also what we've seen from that study, as you hopefully would agree, is excellent clinical outcomes backed by an IRB-approved clinical study. So for that, I'll now pass you back. But thank you very much indeed for your time and for coming out on Sunday, given I know how hard-working everyone is for the other 6 days of the week here in Thailand. So thank you.
Thank you very much, Mr. James. We have a bit questions from some doctors. As many doctors still believe that elimination and provocation diet is a gold standard of food allergy. And how do you suggest those doctors that fish test is more helpful?
So I think I would agree that that is the gold standard, but that's based on having a compliant patient. And I think given patients' busy lives, that I would argue is a much harder thing for every physician to control. And so I think it's very helpful, as we highlighted here, to be able to show physicians and patients, more importantly, that if you eliminate a limited number of foods, then you'll see a very positive outcome in a relatively short period of time. To get a patient to be compliant with an elimination diet over a longer period of time, I would suggest is a greater challenge than to run a food sensitivity test. So I think what you'll see is, given the busy lives that everyone leads, and Thailand seems to be busier than most, that I think it's highly unlikely that you're going to get good levels of compliance with the elimination diet on its own. Whereas if you can augment that by running the FIT test and zeroing in on the key foods of interest that that patient will be sensitive to, then at that point they'll see a much quicker and more aggressive outcome than they would have done by the trial and error element that you see with the elimination diet.
Thank you very much for Mr. James, and please give a big applause to Mr. James again.